Welcome to our DNA project. Today we'll be discussing replication, transcription, and translation. Firstly, we'll be discussing the DNA model. The white styrofoam represents sugar bases. The orange clay represents phosphate. The colored clay represents nucleotides. The yellow one represents cytosine. The green one represents guanidine. The blue one represents thymine, and the purple one represents iodine.
Firstly, we will be discussing DNA replication. Replication is the process by which a DNA molecule replicates itself to produce two molecules of DNA. The idea behind replication is that each DNA strand acts as a template to make a copy of the DNA, resulting in two identical DNA molecules. This is called semi-conservative replication. The original strand is typically known as the parent strand, and the new strand is known as the new strand.
Replication begins with the origin, the point where the hydrogen bonds between two strands are pulled apart by an enzyme called helicase. The replication fork is found at each ends of the bubble, where the strands push open to allow the daughter strands to form off the templates. The bubbles then enlarge and spread out in both directions, and fuse with other bubbles to produce two DNA molecules. This means replication is bidirectional. DNA is copied and replicated in both directions. Creation of a new strand of DNA in the replication fork from the parent template occurs as follows. Helicase forces the strands of DNA apart. Because this often leaves lots of strain on the DNA, DNA gyrase attaches to the strands to reduce strain, and a single strand binding protein keeps the strands apart. Then, an RNA primer is attached to the parent strand from an enzyme called primase. The primer is a starting point for the DNA polymerase III enzyme, which can now begin adding deoxynucleoside triphosphate base pairs. These molecules are similar to DNA, a nitrogenous base pair with a deoxyribose sugar base, but have three phosphate groups, two of which are lost to provide energy for chemical bonding. Then, DNA polymerase I enzyme replaces the RNA primer with DNA nucleotides, and the DNA ligase enzyme reattaches the segment with the rest of the strand.
However, DNA is antiparallel. One strand is from the 5 to 3 direction, while the other is in the 3 to 5 direction. And DNA polymerase III can only work in the 5 to 3 direction. So there are two processes for making the two new strands of DNA. For the 5 to 3 new strand, the process is the same as what we described. It is fast and continuous. This is called the leading strand. For the 3 to 5 new strand, it is discontinuous and slow. This is called the lagging strand. In this process, an RNA primer is moved from the primase, which the DNA polymerase III attaches to and adds base pairs, moving away from the replication fork, resulting in an Okazaki fragment consisting of RNA and DNA. DNA polymerase I then replaces the RNA with DNA, and the DNA ligase combines the fragment to the rest of the strand. A new primer is made and everything repeats.
Transcription information is copied into messenger RNA, aka mRNA. mRNA is a nucleic acid that carries a genetic code to ribosome to produce a protein. This process is similar to that of replication. When an enzyme called RNA polymerase pulls the DNA strands apart, one of the strands contains the gene that produces the necessary protein. This is called the sense strand. Because of complementary base pairing, ATGC, the other strand which will be copied is called the antisense strand. RNA polymerase works only in 5 to 3 directions, so the mRNA chain goes from 5 to 3. The chain starts at the promoter region, the region on the antisense strand that contains the gene. The chain ends after RNA polymerase comes after the terminator, a series of nucleotides that makes the RNA detach and leave.
Translation is the synthesis of polypeptides directed by an mRNA template through ribosomes. A ribosome is composed of rRNA and has a large subunit and small subunit. Between the subunits are three sites: A, which holds activated amino acid to be added to the polypeptide chain, P, which holds tRNA with polypeptide chain, and E, where tRNA is discharged. Each group of 3 nitrogenous bases code for a specific amino acid. These groups of 3s are called codons. Translation is divided into 3 steps: initiation, elongation, and termination. Initiation is when an activated amino acid attaches to the start codon on the RNA on the 5' end. The activated amino acid consists of tRNA that has been bonded to an amino acid through ATP and phosphorylation. On one end of the tRNA is the amino acid. On the other is an anticodon which bonds to the codons on the mRNA. Elongation is when more tRNAs bring amino acid to the ribosome and add the amino acid to the growing polypeptide chain. The ribosome begins to move from the 5' end to the 3' end. Termination is when the ribosome reaches a stop codon at the A site. The ribosome then splits from the mRNA and the polypeptide chain is released from the ribosome.